So yeah, thank you so much for having me back. I spoke at the LabMed 510, the research in progress, about a year ago, and at that point communicated, and it seemed like it would make sense to wait a year or so. A little of this will be a little bit of a reprise of that, but hopefully an update of some of the same general theme. So my disclosures are up here, okay, and so this is where I work, down at UW South Lake Union campus, where we have a bit of a T-cell immune monitoring capability down there, and this is just a little sort of advert to get started. The flow cytometer, rest in peace. We are going to have to sell it because we don't have money for the service contract unless somebody down here is feeling really rich and wants to help us with that. So that one, unfortunately, but we have had the ability to actually do clinical grade immune monitoring as sort of a CRO type function for some vaccine clinical trials. Not really going to talk about that too much more here, but one of my main colleagues down there, Sean, who is home-based in DLMP, as well as Chetan and myself head up this space. So my objectives for today are to sort of review the basic principles of T-cell receptor biology and the specimen types we can use and some of the sequencing platforms that are available, and to understand some of the uses and some of the limitations for T-cell receptor sequencing in vaccine research, a little bit more so than the third bullet point, which is the use of T-cell receptor sequencing for clinical diagnostics. There is one FDA-approved indication at this point for diagnosis of an infectious disease that I'll mention. And then there's the world of monitoring for T-cell lymphomas and for looking for the clonotype that's expanded and is sort of malignant. I'm not an oncologist, and I'm not really going to talk about that that much, but I believe that there's licensure for minimal residual disease, more on the B-cell side for B-cell receptors, but the same general theme, but not really going to talk about that that much today. So this is the sort of arena that we're talking about, or the biology that we're talking about, are specific T cells. The only piece of digital art I've ever paid money for, and I did a little copying on it, where it takes two cells to tango, and the lower cell you could conceive of as a virally infected cell or an antigen-presenting cell that's scarfed up some virus and is presenting some processed viral protein. So that's that yellow peptide there in the middle. That would typically be a viral or infectious disease peptide, to the upper cell, which is the specific T cell. And that molecule that's drooping down in red and blue, that is the T cell receptor that we're going to spend some time on, with hypervariable loops out here at the business end that are suitable for sequencing and specifically for next-gen sequencing or high-throughput approaches to capture the diversity in that population. So, this hasn't changed much in terms of the mechanism of T-cell repertoire generation for the last, 20, 25 years or so. There were some Nobel Prizes on this topic. But just to reiterate a couple of main points is up in the germline, there are many different segments, the V segments, the D segments, the J segments, coming together in a rearrangement. There's actually a ligation event with some sloppy ligation to generate the DNA that encodes the beta chain. There's a similar set of events that take place for the alpha chain is up here, beta chain is down here. And eventually you end up with the heterodimer on the surface of a particular T cell clone. Once that rearrangement has taken place, it's locked in. So this is unlike B cells. B cells can edit their work, sort of the Lamarck kind of type of approach. But T cells, once they've locked in their clonotype, they and all of their daughters and granddaughters and et cetera, multiple generations of cells are sort of barcoded with this sequence that we can use to track cells either over time or over space around the body. And you can imagine both of these chains are hypervariable, and some versions of sequencing will only sequence the beta chain and say that's good enough. That's sort of a hemi barcode, or that's enough. And other more modern single cell approaches will actually sequence the hypervariable region of both chains to get a sort of fully specific barcode. And these regions that are kind of with the brackets here encode this complementarity determining region. So that's the region that then is interacting with the peptide and the HLA molecule. There's actually a couple of these complementary determining regions, but mostly that's the CDR3 is the one that's right in the recombination rich zone. So there's really two kinds of technologies that are now frequently used to sequence T-cell receptors. You can either sequence the DNA or you can sequence the cDNA. And you can imagine there are pluses and minuses of both of these approaches. I'm going to hope that I can find my cursor again. Here we go. So the method that's been sort of commercialized by this company down here really sequences the genomic DNA. And so there's a preliminary PCR where there's a whole bunch of primers that correlate with all of the different V genes, and then there's some J gene primers. You do a preliminary PCR. This particular platform, they actually spike each reaction with what they call an artificial repertoire for normalization. But all of the amplicons then from this region, so there'd be many, many amplicons from a mixed sample, would be, they're amplified with UMIs and additional PCR steps, and then they're actually sequenced, but there's the opportunity to do some normalization for that artificial repertoire that's been spiked in. So for people who were looking for real sort of SOP-driven clinical grade type sequencing, where theoretically one cell leads to one read, because it's genomic DNA based, this one may have some advantages. Much more affordable and sort of either home brew or kit driven is to do cDNA based methods, where your sample turned into mRNA and then some oligo-DT priming. There's a template switching oligo with a UMI, so now we've got each cDNA molecule labeled with a unique molecular identifier. And these are sequenced, and then you can then count the number of sequences with each UMI. So you can get a rough idea of the abundance of your different sequences in this sample. It kind of depends on how many mRNA molecules there are per cell, and if the cell is really activated and making a lot of TCR, there might be more, and if it's a resting cell, there might be less. So that's one of the sort of downsides of the cDNA approach. The cDNA approach obviously is so sensitive that it can be either used for bulk samples or for single cells these days, so I'll show you some examples of that also. So here we are again, and we've got these hypervariable loops drooping down from the T cell and interacting with the peptide and HLA. And maybe a little bit familiar to many of you, and what we actually have for some FDA-approved tests are functional tests of the T cells. So functional tests for T cell specificity. So just a brief refresh on that. And these are also very commonly used in vaccine research. So the mainstream, y-axis on figure 5 in your paper for COVID, figures 1 through 4 are the antibodies, and then they finally get around to the T cells, right? And it's going to be something like ICS or ELISpot or something like that with a functional assay to count the number of cells in the sample. And but aspirationally, we might be also able to sequence the T cells in the sample, let's say after a vaccine, and say something about whether there's been a response or not. And this is based, of course, on this sort of clonal selection theory of lymphocytes, same thing for B cells and T cells, but just sort of a cartoon of the time course that people think about for T cells, is that before you give the vaccine, you've got a naive level of these T cells. It's not zero. So for those of you who are into like obscure things like Gurdjieff and stuff, you're remembering in Plato's, Socrates' dialogues. You were born with everything that you already know, you just have to remember it. Anyway, so you're born with this repertoire, and from a non-zero level, then you get exposed to the pathogen, the levels go up, and then there's a die-off, and they come down, and now they're at a memory level, and you are detecting those cells. Sometimes, occasionally, you see this inflationary issue here. We know this is fairly specific for CMV. So we can actually pick this up in people's repertoires that individual clonotypes are at the 5, 10, 15% level. It's not really your lymphoma invading like MGUS, like for B cells, but it's sort of a thumbprint that CMV infection, chronic CMV infection has on the T cell repertoire for some reason. One idea is that we can use these T cell functional assays as a sort of for memory and to sort of read out somebody's antigenic history potentially. It could either be a T cell functional assay or maybe a sequencing assay. I think you're all familiar with taking a drop of serum or a drop of plasma and doing a highly multiplexed serology. And there's something called virus scan, but we do this for autoimmune diseases too. And you put it down against 50 antigens or 200 antigens, and you can get sort of a readout on all of the antigenic exposures that person may have had. That's high throughput, efficient, cheap. Can we do something like that for T cells too? Is really sort of the question. And when we get into these functional assays, I know I've talked to this audience about this before, but there's a pair of FDA-approved tests for T cell functional assays for tuberculosis exposure. So for either latent tuberculosis infection or active tuberculosis, these tests can't tell. All they tell is whether you're sensitized to TB, Mycobacterium tuberculosis or not. And just sort of showing, this is the only slide on this, that these are sort of our volumes in the DLMP up through the beginning of 2022. We do about 1,000 of these a month. And then we sometimes turn to the alternative test that's shown in the lower panel, which is an ELISpot, which is currently a send out for this test. And these are interesting assays because they're live cell assays. So the cells have to be in good shape and capable of functionally responding and producing, in this case, interferon for this assay to work. So the indeterminate rate is very depressing for our customers. It's about 5%. And nobody likes that when they get an indeterminate result back. Okay. But what about T-cell sequencing assay for antigenic history, for memory? So the first project I'm going to tell you about was started in early 2020 from the push to get convalescent plasmid for COVID-19. So there was a rapid push to enroll people who had recovered and give their plasma for potential therapeutic use. And Anna Wald, who's up top in the middle, was the PI of the local contract, together with investigators from the blood bank, and Stacy, our data manager, Angela, our coordinator. And so people were, enrolled, and if they had high titer plasma, they were sent to the blood bank, and great, thank you very much. But we were also able to enroll them in a natural history study and do some T-cell receptor sequencing on them. And we had a couple people in the department really help take the lead on this, and a lot of correlative work with traditional serology and a couple of different serologies that were being tried at the time. So Mark and Chihiro led that, and then Jim at the Hutch sort of integrated and all the data and kind of wrote it up. So we enrolled these people with recovered from COVID, 302 people, and their bulk blood, and I'll tell you the sample type in a minute, was sequenced. So the repertoire, the smorgasbord, the mix of T-cell receptors in that blood sample were sequenced. And then these were sort of correlated with a public lookup table. So this is going to be a recurring theme here, is what are you going to look all these sequences up against? And in this case, a public lookup table was created. I think I've got a cartoon on how they made it in a minute. For each sample then, a set of numbers could be come up with, a depth and breadth number. I'm not going to go into the formulas for the presence or absence, or really the amount and the breadth of the diversity of T-cell receptor sequences that are thought to be specific for SARS-CoV-2. It could be the spike protein, could be the nucleoprotein, et cetera, et cetera. And so at the end of the day, there was a result for each subject based on a classifier that was built by this particular company, Adaptive. To train the classifier, really a combination of two methods were used. One was a statistical approach, which took hundreds to thousands, I think about 1,500 disease or recovered PBMCs, and several thousand healthy control pre-2019 PBMCs. And bulk sequencing was done on the T-cell receptor repertoires, maybe 500,000 or 1 million cells per sample. And every sequence was statistically compared between the two sets with a massive Bonferroni type correction. And a set of sequences were brought out that were statistically associated with being recovered from COVID-19. The second approach shown in the lower part was more of an antigen-specific approach, in which live PBMCs were re-stimulated with SARS-CoV-2 peptides, and the cells that were activated, and these were sorted with a cell sorter, physically sorted, were then sequenced. And those T-cell receptors were assigned as being probably SARS-CoV-2 specific. And these were sort of put together to make this public lookup table. And there are public lookup tables now with publications for the first three infections shown here, CMV, SARS-CoV-2, and Lyme. Aspirational, and don't work with those guys really anymore. Freebie days are over. Aspirational are some other autoimmune diseases or other diseases where there could be a T-cell reaction against an autoantigen, for example, or a set of bacterial antigens, where it might be possible to build a classifier based on this sort of idea of reading your T-cell memory, reading your immune memory through T-cells. So the sequences were queried against this public lookup table, thought to be SARS-CoV-2 specific, and what did it show? So here's our cohort. It's 302 people. It was focused because these kind of volunteer type people tended to be older, white, with milder disease, and were captured an average of 2 to 3 months after their illness, recovery from their COVID-19 illness. And the specimens were the equivalent of about 5 mils of blood. We used frozen PBMCs, but you can use fresh blood, because all you have to do is make total DNA, and you can do this on the robot. It's nothing special about that. And the averaged around 800,000 reads per sample, so some of them, 85% of them are good, skip the details on that. So we're sampling about 650,000 T cells per sample, and out of that about 200,000 unique clonotypes were identified per sample. Some were superabundant, show you an example of that, a lot of singletons, so there's sort of a gradation from some big standouts down to many things only seen once. And obviously the depth that you sequence to depends on what you started with. It's like many pathology or diagnostic specimens. The more you put in, the more depth and detail you're going to get. So what does this look like over time? What's the reproducibility look like? So here's what a set of 2 samples from one person might look like. Time point 1 on the x-axis, time point 2 on the y-axis. Every dot is a clonotype. A lot of these things are packed on top of each other. And they go from very, very rare on the left to very common, 10 to the -2. So that's really 1 out of 100 T cells way over on the upper right. And you can see that there's very, very good agreement in these 2 time points. So each dot there is a sequence from this person. There are 354,000 down here in this light gray region on the lower left that are too rare for the statistical program to try to say were more or less abundant in one or the other. And the rare ones in red or blue were called out as more or less abundant in one sample or the other. So this is what the technology looks like. When we look inside of a specific sample, here's the top 10 clonotypes, and that big boy on the left is 11%. That might be a CMV-specific clonotype, but then they rapidly are going down to the 1% level, and there's a giant tail of very rare clonotypes that are coming off on the low end. What does the raw readout or line listing look out? Just sort of a snapshot. It looks kind of like this. This is for the beta chain again, and this is that CDR3, that loop that's dangling down right here and touching to the peptide. Always starts with a C, always ends with an F, and it's 10 or 12 amino acids long. There's 20 different amino acids. There's a lot of combinations in there. So that's kind of the barcode, or you can use the nucleotide version of the barcode. But this is what the computer is now going to be looking up against your lookup table to see whether you've got sequences that are associated with your infection. And so the punchline of the results was that the T-cell sequencing assay was a bit more sensitive than some of the serology assays. And we had the Euroimmune, which was a S-specific. The Abbott Architect was an anti-N, and then the neutralizing antibody assay. And you can see by and large there was pretty good agreement with the T-cell test. There was a little bit of disagreement where one of the assays was more or less sensitive, one or the other. This was published about 2 years ago, and I'm not going to make a big deal about this, but wanted to show this as an example of this use where the performance was reasonable. Actually, I believe that the company got an indication for this test. I don't really think anybody uses it that much. I mentioned there were 2 parameters, the breadth and the depth, that you can pull out of this. And as the neutralizing antibody titer went up, these parameters for based on T-cell sequence reads also tended to go up, sort of correlating maybe with the intensity and time of infection and antigenic challenge. The CD4 levels, these were T cells that were assigned to be CD4, and I'm not going to go into the exact details of that, but they were better correlated with the neutralizing antibody levels than the CD8 assigned T cell receptors, which is consistent with our concept of CD4 T cell help to B cells. There is likely to be a better correlation between those two parameters. And we knew this a couple of months into the pandemic, that if you were male or sicker or older, you had higher antibody levels, more antigen, more immune response. That also seemed to be true for this T-cell response. So comparing hospitalized and unhospitalized, people reported a history of fever or no fever, reported shortness of breath and so forth, or were male or older. All of these things were also correlated with a more intense, if you will, T-cell response as read out with this public lookup table. And in terms of durability, this was early data. The anti-S and anti-N levels were sort of drooping with time. The red lines are the non-hospitalized. Things looked a little bit flatter for the T-cell data, but this was all passé after about December 2020 when everybody started getting vaccinated. So now we're in an era of hybrid immunity. So I think I'm going to switch now to T-cell tests in the current era of hybrid immunity. Either you've had the vaccine only, if you're lucky, or you've had COVID and the vaccine. And so, or you've had breakthrough. So we're in that sort of era now. And we were able to extend this study actually with nice help from Anna through about a 2-year period. So this is at the beginning here where we were studying these early samples. But then people got their shots, and we got some more samples, and we were able to study, before, during, and after, the shot 1 and shot 2. And then about a year later, if you guys remember, it was about December 2021, January 2022, we all started getting boosters, and we were able to get, typically just after the booster, and able to get a nasal swab also at that time to study the localization of T cells to the nose, respiratory, relevant site at that time. And this is just a table of events. It's about a 2-year period, about 600 days. The black squares in the middle, or the black symbols, are where people got vaccinated. And the triangles, I think, over at the end, are the samples that we got. The black squares over on the right are when they got their boosters. So swimmer's plot, whatever. A lot of effort, a lot of samples, 53 people overall. And we had great serology support from Anton Scholich over at the Hutch, who had a Luminex or microbead-based multi-antigen ELISA. So he studied all of these samples, hundreds and hundreds of samples, for both anti-N and anti-S over this 2-year time period. And now they're centered. Day 0 in the middle is the day of your first shot. So go backwards in time to those negative numbers where people were sick, and then forward in time is to the end of our study. So, this sort of is, I think, super cool, and, speaks for itself that everybody was positive for anti-N, and, they sagged. And then when you got your S-based vaccine, they didn't go up. You know what? Because there's only S in the vaccine. There's no N in the vaccine. But what about these 3 people? Well, they had a breakthrough infection. Okay, so their N went back up. This person here came to the clinic and said, I had COVID. I want to give plasma. Look at me. But, it looks like they never had infection. So there was the same person who was negative for both anti-N and anti-S. But when they got their shot, their titer went up to have anti-S, but it didn't go up that much. And then very nicely, everybody's anti-S went up. Okay, so we had the opportunity to do some T cell studies longitudinally over time in this extended cohort at this point. And we did a little functional stuff too. I wanted to show Shira because she's, I think, applying for path residency and was a medical student with us last summer. And she did some functional tests before and after the vaccine. So the little syringes on the lower right are the vaccinations, and we had some stored PBMCs showing. And yeah, sure enough, these were responses to S. They were present, so these are non-zero ELISpot responses. Here are spots, cells per million cells in the PBMCs for S. And when they got their, shots, the responses went up. So this was nice, but pretty, well established. So the objectives of this study were really to look at the kinetics of vaccine boosts after infection and try to compare CD4 and CD8 T cells. And we're trying to try to create really more personalized lookup tables and not rely on this community lookup table, which has noise and has problems that I didn't really go into. And also to see whether these TCR sequences clustered with each other, because they're complex, but they're not infinitely complex. And can we start to see some structure, community structure, when we look between people? And then finally to look in these nasal samples and see if there were tissues there, samples there. And so here's kind of one of the punchlines of the whole talk, where here we're comparing somebody before their first mRNA dose and then after their second dose. So this is about a 7-week period or something like that. And all of the little black dots that are lining up on the XY axis, those are clonotypes that are unchanged in abundance. But all those red guys, and some of them are up 100 to 1,000-fold expanded, are candidate SARS-CoV-2 specific T cells, each dot being a specific clonotype. We haven't proven anything yet, but we compared the before to the after, and they went up like crazy. So what can we do to kind of access those a little bit more? There are candidate spike-specific vaccine-responsive TCR sequences. There's hardly any blue ones that went down, and there were 620,000 low abundance ones that were excluded from analysis, the rare ones down there. So we turned to next-gen sequencing to try to get an overlay of an orthogonal data set to try to work on this. And I know that DLMP very generously placed some 10x chromium controllers at several sites around the UW Medicine. We didn't actually use one of those. We had access to another one that through dermatology. But after paying our tuition, we got this working. And what we would typically do would be to take some peripheral blood mononuclear cells and dump in spike peptides. And after an overnight incubation, these 2 markers here, CD69, CD137, so-called activation-induced markers, we would sort the cells that turned on after re-exposure to the spike peptide pool and put those through the platform. And we decided, so we did our paired TCR alpha beta sequencing. We did some tagging with oligo-labeled antibodies to try to, phenotype these. Are they CD4 or are they CD8? We kind of blew off the RNA-seq. We got it on a couple of samples, but you know what? These cells have been kicked like in the rear really hard for 18 hours in tissue culture media. And how meaningful is the RNA-seq pattern going to be in those cells? I don't know. It worked technically, but we didn't make a big deal out of trying to look for subsets or clusters or activation gene signatures or anything in that so far. And the slide on the right here is kind of the overlay punchline. So on the left, you're seeing the before versus after. And on the right now, we've overlaid it with sequences that were also identified by putting in the spike peptides, sorting the reactive cells, and then asking, do they match or not? And by 6, this is 2 to the 6th, and that means we got, I don't know what that is, 32 copies of that gold one. We actually, by the single cell method, this clone, sister clones, was recovered 32 times. So you can kind of follow the rainbow and see that there's a nice correlation between the fold increase just by the total blood sequencing and the frequency of recovery by the single cell platform. So to us, this kind of confirms that this relatively low-tech whole blood assay seen on the left, which does cost money, but it's just sort of a 5 ml tube of blood, can give you some clues that you can kind of validate using or with a single cell method. And it's interesting because some people got bang out of the first shot and bang out of the second shot. So here they are bridging after both shots from pre-shot 1 to post-shot 2. Other people really didn't get much action out of their first shot, and they got this clonotypic expansion out of the second shot, and it was verified by the single cell platform. So if you summarize that for pre-shot 1 to post-shot 2, these clonotypes here were all unseen before the vaccination. So everybody was a little bit different, but that's what we're trying to access, I guess, these days with our platforms, not have a one-size-fits-all. And when we phenotype these guys for CD4 versus CD8 using the antibody oligo-labeled antibodies, what we found was that, I'm just showing an example here, is that most of the spectacularly expanded clonotypes happen to be CD8s. And in the long arc of vaccinology, we got vaccines that make antibodies. We got vaccines that make CD4 T cells. You kind of have to have CD4 T cells if you're going to make good antibodies. But a vaccine platform that makes good CD8s, we've been waiting for that for a long time. And now we've got that for mRNA. Arguably the adenovirus platforms and the poxvirus platforms are also kind of in the same kind of area. So we summarized this here, and I don't want to belabor this too much, these sort of complexities, but in this graph here on the left are 5 individuals, and each line is a clonotype tracked over time. So, and it's a log axis. You can see with the onset of the vaccination here in the middle, many of these CD8 clonotypes go up, 1 to 2 to 3 log 10 in terms of their abundance in the whole blood. So now we're back correlating ones from the single cell platform and back to the serial whole bloods. While in contrast for the CD4s, we're not seeing that much in the way of an actual liftoff or expansion. And this is summarized here on the lower right, where this is the mean S-specific clonotype frequency in 15 people who were tracked over time and over their sort of cartoon here of their time of events, where we would typically get a 2 to 3 log increase in the CD8 T cell abundance. Now this is, hybrid immunity. You're already immune. You've already got memory cells floating around in your body. To be like this, and we've not been able to do this yet for naive people who just got the vaccine but had not had previous COVID-19. Okay, so what about a little bit more about the sequence structure here? And in this case, we had 24,000 cells that passed through our single cell platform, and what I showed you before, we were using people as their own lookup table. Right? We were correlating their own single cells to their serial blood. Now we're going to sort of throw together all of these single cell sequences and see what it kind of looks like. And we're going to include in this look here the ability to cluster T cell receptor sequences if they're almost the same. So if the alphas are identical and the betas have like a glycine to an alanine substitution or some sort of very conservative thing, we'll allow those to cluster together using an algorithm put together by somebody whose picture I hope I'm going to show you in a minute. Hope I left it in here. But this is kind of what the structure of that looks like when we perform the sequencing. So 5,700 cells participated in clusters and formed 1,400 clusters. Out of the 24,000 cells fell into 5,700 clonotypes, and 1,400 of those fell into clusters with at least one other T cell receptor, forming finally 284 T cell clusters. And when you make a map of those where the edges or the lines connect clonotypes, every dot is a clonotype, you start to see some structure. We used TCR DISC3 by Kashmeyer-Blackwell, who's at the Hutch, as the algorithm, using the paired data, so data from both the alpha and beta chains. The distance parameter that you allow for clustering is tunable, and we set this to be very conservative, so the sequences had to be almost the same in order to be forming a cluster. And it uses this BLOSSOM62 matrix, which many of you know probably better than me, which gives gap and substitution penalties that's sort of biochemically aware. But you can also tune the matrix as you want. So when we start playing around here and putting different peoples as different colors, we see a lot of these clusters contain components from different humans. So they're populated by T cells from multiple people. And let's zoom in a little bit on this now. So a couple of features pointing out in this zoomed in region. If it's a public cluster, it's a dotted line. I think I can see that it's sort of masked up top. But there's a lot of dotted lines in here. So the dotted lines connect clonotypes from different humans. The size of the circle depends on how many sister cells came out of that person or that are identical. So there's a lot of big clonotypes in here and then some little ones. The relative position of these clusters to each other doesn't mean anything. Some of them they're color-coded by CD4 or CD8, and we would expect a cluster to be either all CD4, like 23 or 20, or all CD8, like 5 and 4. That would be expected biologically, and that largely seems to come true. And actually, prior researchers had found some clusters that were associated with HLA-A2, common type, and for which the peptide was known, and we found those again. So we thought that was fairly good reality check. But in addition to finding 1 and 2, we found a ton of other ones. Focusing on 0 and 1, focusing on these ones that were previously known, let's now look, and each line is a human, let's look at the summary abundance of T cells in that cluster over time. And this makes sense. So here they just recovered from COVID, E00. Now a year went by to E01. Things were sagging because a year went by and memory was going along. And then they got their vaccines, and the abundance of those clonotypes went up in the blood. So that, sort of was another reality check we thought that made sense. But here we're again looking at these known clusters that had already been, determined. So what about previously unseen clusters? So here's some previously unseen clusters 4 and 5. And these sequence motifs, this is the alpha chain, this is the beta chain, and these are the sequence motifs for that complementary determining region, that hypervariable region. They also have these nice kinetics by and large, where they sag down and go back up after the vaccination. We decided to test these by T cell receptor reconstruction. So you're all familiar with making monoclonal antibodies by taking single B cells and sequencing them and manufacturing the antibody. Well, we can do the same thing, take the single C cell, sequence the T cell receptor alpha and beta, make artificial T cell receptor, make artificial T cell, and test it. And this is one of the examples we did here from this cluster 6 here. There was participation in this cluster from 7 out of 7 people who were HLA-A3. So we thought it was probably restricted by A3. Had the nice SAG, had the boost with the vaccine. And sure enough, so I'm going to skip the details here, but the reporter cell has a fluorescent reporter in it when the T cell turns on. And if we put in the individual's 4 HLA cDNAs into an artificial antigen presenting cell, so it's artificial cell versus artificial cell, but we put in the individual's 4 different HLAs in without spike, nothing happens. But when we put in A3 and spike as a transient transfection, we get a nice turn on in this cell, sort of validating that, closing the circle or whatever, that we found a spike reactive T cell by doing this. And I thought this was really cool because there's a rule breaker in here too. Some of these sequences came up in these clusters 2 and 3, but sort of a rule breaker, like they didn't really sag with time and then go back up. They just were sort of high the whole time. And when we started looking at these sequences and these sequence motifs, they're actually already known as mucosal, I don't know what A stands for, maybe somebody does, invariant T cells. So these are innate spectrum T cells that hang out in barrier tissues and can be activated by cytokines and by non-TCR signals. And so we found these in there. They turned on during this peptide stimulation shown on the upper left. And again, I've lost my cursor already. But they were sorted because they got activated. And the hypothesis kind of is that they got turned on in that cell culture pot by interferon and other cytokines that were in there. But when you think about the in vivo situation, actually innate cells, these innate spectrum cells, have been implicated in both protecting experimental animals against certain infections and also immunopathogenesis. And in fact, if you have severe COVID and you're in the hospital and you get your blood drawn, and a lot of these studies were done in 2020, your innate cells are turned on like crazy. It's kind of part of the cytokine storm, sort of immune generalization kind of thing that can go on if you've got high levels of cytokines going on. I think we saw this in vitro here also. Okay, let's see if I can make this work here. All right, so we had the opportunity to put Q-tips in people's noses at the end, and we wanted to look for potentially protective T cells at the site of your next inoculation when you next sniff in COVID-19. We actually just did these flocked swabs and actually put it into some RNA save buffer. And then we realized that we actually wanted to change stream. And so we made DNA anyway, and it worked. I forget, Li Chen Jing did this in the lab. And submitted that for the quantitative T cell receptor beta sequencing. And the graph there just kind of shows probably the blue line is the more relevant one, is sort of the number of productive TCR templates, which is sort of correlates with the number of T cells per sample. There was quite a range between 200 and about 50,000 T cells, equivalent T cell genomes recovered per swab. If you spend any time around kids or snot or something, you know that everybody's is a little bit different and everybody's nasal swab is going to be a little bit different. But we were able to get T cell receptor sequences off of these samples. And when we correlate this, a couple of different lookup tables, yes, the T cells are there. So in this particular case, this is the patient that we made 6 individual artificial T cells from. And the T cell receptor sequences in their nasal swabs are ranked from the most abundant to the least abundant. So there were actually 600 different T cells recovered from this person's nose. And all 6 of the ones that we proved using blood and that other pathway as being COVID specific were actually also found in the nose, and some of them are quite specific. So this was 1 out of 100 T cells in the nose, was this exact clonotype here. And similar tables from another 12 people are shown here. In this case, we were using personalized lookup tables, and that was based on those sorted the sorted single cell sequence to create these personalized lookup tables. I know it's a little small, but you can see a lot of blue dots sort of somewhere along these, I forget what they call these curves, but somewhere along these curves where there is some population of documented COVID-specific cells. We used this other personalized lookup table method here where we just took the T cell receptor sequences that were in the after and not in the before, mapped those back onto the nasal swabs, and everything in red here was a hit from that method. So again, using the personalized lookup table approach. And then finally, there's a community lookup table. In the last 2 slides here, I'm going to show you some work now tracking all the way back to that community lookup table that you can download version 1 from Adaptive for free, but version 2 is secret and you can't download it. So using the publicly available version of that, there's 4 people shown here. This person was followed over time between 2020 and 2022. They got vaccinated in there, and look, they're getting more S. The x-axis here is across the genome of the virus. They're getting bigger blobs here against S as time goes by. Same thing with this person here. They're getting this big clonotype here. But if we look in the nose and just using sort of the public lookup table for the TCR sequences, this is again after their booster, we can see some sequences there. Summary, 15 minutes to go, great. So T cell receptor sequencing in vaccine research. T cells undergo clonal expansion and persist in the blood and tissue, and that's kind of the basis of this whole hypothesis or this whole method. the T cell receptor data space, if you will, it's large and complicated. We probably have about 10 to the 7th memory T cell clonotypes in our body, but it's approachable by next-gen sequencing or single-cell modes, especially if we pre-select the single cells that are going to be likely to be antigen reactive with our antigen of interest. And the sequences can serve as useful barcodes for expanded clonotypes, tracking either within person over time or within space within the person. So it's a potential horizon for querying people's past or present, antigenic history. It's limited by expense, and these assays aren't fast either. And mostly the availability of the lookup tables, those are still really quite limited. And the training data that would be used to create these lookup tables, it depends on HLA diversity. We didn't go into that in detail, but I think you can kind of intuit that. It depends on what HLA alleles are in your subjects. So that's going to thus correlate with geography and ancestry of where your training data is coming from. So a training set that was created in one continent or one group might not be relevant to another, as relevant to another group. I think that these analyses are enhanced by this fuzzy matching. I think we've proven that the fuzzy matching is valid, especially if you use conservative parameters to group the T cell clusters. And the last point is just sort of rehashing what I said. And I don't have a picture, of Emily Ford or Koshmeyer-Blackwell, who kind of led the single cell work. I had to put in a little bit older picture at the last minute, I apologize, but many people in the lab have helped. Li Chen did all the wet lab work with the single cell work. Carrie did a lot of work sorting memory and naive cells that I didn't show you. Chris is hugging the flow cytometer. Anna led the specimen collection. Anna Wald at the clinic led the specimen collection work for these studies with help from Christine Johnston. Amalia with the statistics, and Kirsten kept us honest with the IRB. Over at Fred Hutch, Koshmeyer-Blackwell, and then Andrew Fiore-Gartland, another data scientist. He has a background in nuclear physics. Helped with the high-dimensional data, and Anton did all the serology there that I mentioned. All of the neutralizing antibody assays, and there were hundreds and hundreds of neutralizing antibody assays done at BSL-3, were done by Mike Holbrook at the NIH and a large team of technologists there. And that's what I got for you. And there's my evaluation, but I'm so old, evaluations don't matter. Keith.
So David, you kind of blew off the mRNA-seq on your cells, saying, oh, they're stimulated. But I keep thinking back to like the HIV vaccine studies, and people look for polyclonality, and they think that there's something about the quality of a T cell that's been generated. Right. And I think all those are done on stimulated cells. So I mean, is there a potential story there?
No, there is a potential story there.
You basically got the data for already.
Well, when you start getting down to nuts and bolts of lab work, each library costs money to sequence. So we bled some money for the TCR libraries and for the antibody tag libraries, and then are we going to bleed another thou each for the RNA-seq libraries or not? So for this particular project, we decided not to do that. But higher level, you're absolutely right. Studying the potential of what a T cell can do when it gets activated is of interest. Am I going to make, interferon gamma, antiviral thing? Am I going to make IL-4, 5, 13 and be an allergic cell? That is, I think, a current horizon, is trying to see the biology through the noise that we now have the ability after we stimulate cells. I know many people do research where they put chemotherapy drugs or other manipulations on cells in culture and use RNA-seq, and it's valuable and interesting. It's just hard to know how to evaluate that for this kind of primary T cell work. Yes? Brian, go ahead.
So, are there any of these T cell types that are associated with long COVID?
I'm not an expert on that. I saw some interesting research from Emory at a conference last week where they were using fluorescent spike protein to count the B cells that were reactive with spike in the blood of individuals with PASC, long COVID. And they were finding that in your routine recovered person, they were, 1 in 10,000 or 1 in 100,000 kind of nice memory T cell levels. But in these patients with PASC, they were 10 to 100 times more common. So there seemed to be a prolonged activation of the B cell compartment in those individuals, and I can't give you the details on this. The hypothesis by that investigator was that if you're going to have all these happy proliferating B cells in the blood, there has to be antigen somewhere. So it's like, okay, now where's the antigen? Is the virus in some sequestered place and replicating? And there's not a lot of energy on that. I think you're asking a really interesting question about the phenotype of these cells. So to get at these phenotypes, and correlating with Keith's question, to get at their phenotype directly ex vivo when they're resting, you have to have tetramers, and then you have to sort the rare tetramer positive cells, and we could do that, and that's work. Is it of interest to take those people with long COVID, hit their cells for 18 hours or 6 hours or something in the test tube, wake them up and see the flavor of how they get happy, and then compare that from the long COVID to the routine convalescent or not? Might be. Susan.
So I have my own question, but I'm going to, Mark Wenner asks a question online that very much follows up on that, but kind of expands, looking at long COVID and differences in TCRs, but also correlation with autoimmune markers, because there is some literature that is debatable about autoimmunity for acute or long COVID.
Yes. I think maybe that's sort of a two-part question. I think Mark may be wondering, are there T-cell markers for autoimmune diseases? That doesn't seem to be the same question, but I think that's a horizon for this kind of work. For example, citrin-related peptides in rheumatoid arthritis, there do seem to be families of T-cells that recognize these citrin-related peptides, and T-cell receptors for those are starting to come into focus there. I think what Mark is referring to is that patients with severe COVID have a large variety of autoantibodies, anti-cytokine, anti-interferon type 1, anti-interferon receptor antibodies. And I'm not, and maybe Mark can tell us, that familiar with whether those antibodies last into convalescence and into into long COVID. But I think that the opportunity to probe the repertoire of people with long COVID would include, if we had the lookup tables, if we had the resources to look for T cells that recognize autoantigens as well as microbial antigens.
Yeah, and then my question, which was totally different, is I love the idea of looking in the nasal swabs for mucosal T cells, and I wonder if there's any clonotypes that you found that might be signatures of mucosal T cells, or if they're really reflective of what's in the circulation.
So these mate cells, I believe, we should get like Chetan Seshadri, or maybe we've got other people who were experts in these. Some of these innate spectrum T cells do preferentially hang out in mucosal tissues, the ILCs, and please somebody speak up if they know more than I do. We've actually not looked at these nasal sequences that thoroughly yet for those signatures. Dan.
David, you remember this? This might raise some PTSD for you, but remember once upon a time we were looking into like chronic fatigue.
I knew you were going to say that.
And just, the long COVID thing reminds me a lot of sort of the chronic fatigue that we were trying to use very primitive tools to try to understand a long time ago.
Right.
And so, is there anything come up?
Well, I think,
new technology.
yeah. I mean, believe it or not, we had a hypothesis in that grant that we were going to use the primitive TCR sequencing that was available back in the day. This was like 20 years ago, which were these like spectra types that, right? And look for a common TCR that was abundant in people with chronic fatigue and then figure out what the antigen was. It's now becoming increasingly possible. If I have a T cell receptor of interest, and this was done to a brief segue into a case of post-anti-PD-1 fatal encephalitis. So, after people get PD-1 blockers, they get a variety of autoimmune diseases. There was a patient studied back east who had a fatal encephalitis, and there was a T-cell receptor sequencing done in their brain, and an abundant clonotype was uncovered, a CD4 clonotype in their brain. That was blasted against the community lookup table and was an EBV-specific TCR. And then individuals went back and researchers and studied the brain with EBV-specific probes and found there was a lot of EBV replication going on, and felt that this was both EBV reactivation but also an over-enthusiastic or immunopathologic response against EBV there. So there is maybe a model for using TCR sequence data in an inflammatory condition of unknown etiology, going to the TCR sequence and either looking it up in the community lookup table, or if you're really ambitious, you can make the artificial T cell with that heterodimer and sort of library screen the universe. And we could talk more about that if you're interested, but there are some very high-end labs that can do library screens against the peptidoverse and kind of figure out at least some things that TCR can see.
Can I follow up on that question? Can you model that instead of searching?
So yes, there's a guy at the Hutch, Phil Bradley, who recently had an eLife article about this, and that's on the horizon. So you get this sequence and of these two loops that are only 10 or 15 amino acids long, and using both solved crystal structures from the database and AlphaFold, and he implemented a custom implementation of AlphaFold, I think, for dealing with heterodimers, because the core AlphaFold is for single chain proteins, as I understand it, and was able to predict in silico the specificity of a few very well-studied TCRs by sort of half-blinding himself to what they saw beforehand. We've actually tried that with some of these COVID TCRs, and so far it's been sort of a fail, but it's aspirational, I think, and for a family of computational structural biologists to try to do this. Kelly.
I was sort of curious about your thoughts for the lack of CD4 responses in the patients that get immunized. I mean,
right.
They make antibodies, and I sort of thought that...
So it wasn't really a lack, and we saw on average a 2 to 4-fold increase in clonotype abundance for the documented CD4 clonotypes. It was, and maybe, we're not sampling their germinal centers and their, lymph nodes and where the action probably is for getting the antibodies juiced up. Yeah, so, one of the things we always have to do is raise our hands and say, there are 2% of the lymphocytes in your body are in your blood. So we're, except for this nose swab, a little bit blind to what's going on everywhere else. Great. Well, we're coming up on the hour. Hope you've enjoyed. Thanks for having me back. I think this is my third time doing this over the last 30-ish years. So Keith, thanks for coming. Thank you all for coming. Appreciate it.